Welcome back to the afternoon session of the meeting. So it is my pleasure to introduce the next speaker, who is Christina Cobbold. And Christina is a professor of mathematical biology based at the University of Glasgow since 2003, and currently holds a Leverhulme Trust Research Fellowship. She's an associate member of the Institute for Biodiversity, Animal Health and Comparative Medicine, and is an active member of the Boyd-Orr Centre for Population and Ecosystem Health. Her research is broad, but generally she involves applying mathematical modeling tools to understand processes in population and evolutionary ecology, with a particular interest in host parasite and host pathogen systems and how these are affected by environmental changes. So I think we'll hear some of that research today. So over to you, Christina.
Thanks, Emiko. Right. Okay, that's for the mosquitoes coming. So that's what these are. If you're wondering what these guys are swimming around, they're mosquito larvae, and then these things at the top are pupae. Well, first of all, thank you very much for the invitation. It's lovely to be here today. Sorry, I shall come around over here. So thank you very much for the invitation. It's really nice to hear quite different talks to things that I normally hear, and so I found them really interesting. So the work I'm going to talk about today is a little bit different to the kinds of things that you've heard so far, in that I'll be talking a bit more about population responses to the kinds of climate changes that we've heard about. So before I actually get stuck into the science, I'm going to thank people so that I don't miss that, because that's super important, because so Dom Brass and Ben Fellows did basically the most of the work that I am going to show you today. So I think it's really important that I thank them before I do anything else, because the credit is theirs rather than mine. But I also want to thank my various collaborators, in particular biologists that I work with on this project. So the kind of populations I'm going to talk about today are insects, and there's lots of reasons why I'm particularly interested in insect systems, but they have very complex life cycles, and so because of that, they tend to have very rich dynamics. But also they're cornerstone species for our ecosystem, and they're involved in many parts of our ecosystem and ecosystem services. I'm particularly going to talk today about their role as vectors for disease. And so this is an example of one of the mosquitoes I'll be talking about, but I'll be talking about other insects as well. So how does insects and climate come together? So this is a sort of caricature of a typical life cycle of an insect. So most insects broadly go through these 4 life stages. There's lots of added extra complexity that then gets put on top of that. For example, this stage can be broken into many other parts, but this is the broad picture. So we go through eggs, larvae, which are like those swimming things, for example, that you just saw on that video, or like caterpillars, example of a larvae, pupae, think cocoons, and then adults. So there's, for example, an adult mosquito there. And so the thing about insects is they're ectotherm, so it means they're highly sensitive to the environment that they're in. And so all of these processes that these stages are involved in are sensitive to the environment. So, for example, the time it takes to go from egg to larvae is dependent on things like temperature and precipitation. The chance that they're going to actually survive that life stage to age depends on these things. Things like land use and the availability of food resources. All of that can then also affect stages like the larval stage. So and affects things like habitat availability for these. So there's lots of ways that the environment affects this system. So they're very, very sensitive to the environment they're in. So then if we think about that in the context of things like climate change and that we've been talking about today, then here's some examples of some current predictions for trends in temperature and precipitation over the next century. And what the thing to take away from these maps, the sort of red and blue areas, is that we're expecting some change, some change in the positive direction for temperature, for example, some in the negative direction, but current projections are projecting some kind of change in the environment. And we're already seeing insects respond to that, and it can do various things, like it can change the seasonality of insects a lot. It can mean that they emerge earlier in the year or later. It can affect where you find them. And all of these things have knock-on effects then for the population as a whole, and then also all the other species that then interact with them. So I'd like to try and unpick this a bit. So we might think ahead of time, right, well, this insect's really sensitive to climate. We're having climate change, so, you know, we're going to see big changes. So the question is, that's going to be the case? Well, insects have evolved in a changing world, right? So if you think about daily temperatures, they fluctuate, right? Through the day, we have seasons. So they've evolved in an environment that's kind of messy and noisy. And so they have their own strategy for coping with this. So, most insects, and in fact, a lot of organisms in general, can do something called phenotypic plasticity. So what that means is that, so a phenotype is a collection of traits. So, for example, it could be body size, fecundity, your survival in a particular environment. Those are all examples of traits. So for us, for example, like hair type or something like that. And so that collection of traits is an individual's phenotype. And so what phenotypic plasticity means is that they can change their phenotypes. They can change what traits that they're displaying in direct response to the environment. And this isn't a genetic response, so it's not because they're evolving. It's an actual real time within a generation type response that you can see. And these pictures illustrate some of these. So these aren't different species that I'm putting here. This is the same species, but it's showing this phenotypic plasticity. So I'm going to just show you this example at the bottom. It's quite a nice one here. So the left, I think, is females, and the right is male grasshoppers. The top 4 are from one location. The bottom 4 are from another location, 15 kilometres apart. Okay. And the top ones, the area was very dry. The vegetation was really poor quality. The bottom 4, the vegetation was really lush. There was lots of rain, really high quality food, and you can see the difference in the size. And so one of the consequences of these larger individuals in this better quality environment is that they're then producing more eggs, they're going to have higher survival rate, they can fly further. So all these things are coming along with that. But this is an example of phenotypic plasticity, because the next year, these 2 sites, in terms of their environmental conditions, switched, and the kinds of grasshoppers you saw also changed. And it wasn't because they moved, it was just because of an environmental response. So insects can cope with a certain amount of change. And so we wanted to kind of think about that in terms of populations and put it into population models. So that's what I'm going to be talking about, is like, how do we include this kind of individual variation? So it's mechanistically driven individual variation. So it's not due just to environmental noise, but there's some kind of biological process that's causing the variation. How do we put that into models? And then what does that tell us about how populations are going to do under climate change? So let me start by introducing you to the framework that we came up with. Before I take you down to the actual sort of equations, let me explain how people currently think about this. So because there's a lot of literature in the ecology literature on phenotypic plasticity. So people typically in the lab measure these what's called a reaction norm. So what they do is they rear the insect or whatever population they're looking at under different environmental conditions. So, for example, different temperatures, and then they measure the trait of the individual. So they could measure the fecundity of that, or how long it takes to develop through a particular life stage. That's the kind of thing they measure, and that gives what's called a reaction norm. And then people use that to try and say something about the population. So the kinds of things they do are, for example, they might take two different species, get the reaction norm for two different species, and then use that to kind of say, oh, okay, well, maybe in this environment, species one might do better than species two. This environment here, it might be the reverse. So that's the kind of comparisons they make. People also might look at a single species, but then look at the distribution of traits in two different environments for that one species. So that's a bit like, for example, that grasshopper example that I just gave. So look at those trait distributions. At the level of thinking about population models, then what people do is you can actually put this reaction norm into your equation. So if you've got your favorite population model, and it might have a growth rate term, for example. So if you've got some ODEs, for example, you've got a constant that represents your growth rate, you might want to now make it environmentally dependent and use this reaction norm to parameterize it. And then if your temperature, for example, is varying as input into your model, this reaction norm would then tell you how the growth rate was changing. So those are examples. And so this is good, but one thing it's sort of missing is that it's sort of coming at things very much from the perspective of this is an individual response to the environment, and there's feedbacks that this thinking isn't taken into account, because some of the environments that traits respond to are dependent on the population density. So one example of that is food availability. The food availability for example, a larval stage, it's going to depend on how many others are in the fighting out for that food. And so in that way, there's a feedback between population dynamics, traits, environment, that this thinking is missing. And so we wanted to include that into our approach, and to see if that could also help us unpick some of the kind of conflicting things that people were finding in the experimental literature of when they were trying to determine, well, is plasticity going to be beneficial or not for populations? So this, in terms of the mathematical framework, this is a bit of a sketch. So if we just forget about the coloured flow diagram, just look at the white one on the left, where it's just going vertically down. This is typically how people model stage-structured populations. So they would partition the population into the life stages, so egg, larvae, and so on, and then you would track how much of your population are in each life stage over time, and you would work out the transition rates between those. So let me just kind of show you what a typical model would look like for that. So there's that flow diagram. The environment's affecting these various rates, and basically, in simple terms, it's we look at the rate of change of our population in a particular life stage, and that's made up of recruitment into the life stage. So for example, recruitment into the larvae comes from eggs. Maturation out, when you've made it through the larval development, you're on to the next stage, and mortality. And then you can unpack those and start putting in your details. I'll unpack them a little bit just to kind of give you a bit of an idea of how things work. So maturation then is going to be, it should be what comes out should be the same as what goes in, but you account for the fact that some of them might not have made it. You might have died along the way. So S is the probability you actually survived your period of time in this class. So if you've survived, then what your maturation and recruitment should be the same. Okay. But then we also have this delay here. So these models are delay differential equations, and that delay accounts for the fact that you have to spend some period of time in a particular life stage before you can move on. You have to spend some time as an egg before you've done all the various biochemical processes to become a larvae. And these taus are accounting for that. And those things can depend on the environment. And indeed, quite often they're temperature dependent, for example. So we can write equations for all that. I can write down the equation for the survival and for tau. And to complete the model, we then also need to write something down for what happens the recruitment right at the beginning, which is where the births come in. And we look at all the life stages that could produce offspring, how many are they producing, and that gives us our recruitment. So that's the basic building blocks. So this is the kind of typical stage structure modelling that people do. We've then extended that by partitioning each life stage, so that's what's going on here horizontally here, according to the phenotypes of the population. So rather than explain the general model, because it's kind of looks quite messy to write down, I'm going to illustrate it through by actually looking at an example. So, and the example I'm going to show you is for Nicholson's blowfly. So Nicholson was an ecologist around in the 50s, did some amazing experiments. And this data set that I'm going to show you is just incredible. It's one of many that he did. And so here's one of his experiments. So the dashed line is the data. He was monitoring the adult population of blowfly over time, and he changed the food during the experiment. And they were counting each individual. So it really is incredible data. And so what you see, if you look at the dashed line, when the food was high, we've got some oscillations. They seem to stabilize a bit once food has been lowered. And the other thing that's happening is as food is lowered, the population has gone up, right? Which is not necessarily what you would expect if you had to guess ahead of time. And so in fact, Nicholson had a hypothesis for that. He thought it was due to phenotypic plasticity. So we thought, haha, right, we can test this. Let's see if we can use our framework on this. So before I show you what we came up with, I'll show you a model that doesn't have plasticity. So Bill Gurney and others in the 80s did a great life cycle model of the blowflies. It was really well parameterized, really carefully tied to the biology. And the solid line is the output of that model. And what you can see is as you lower the food, the population goes down, and also those population cycles they're not really stabilizing. So you're not seeing what was seen in the data. So it seems like if not having plasticity, there's something's missing. It's not enough to explain it. So could plasticity be the answer? So that was where we started. So where is the trait variation in blowflies? So there's two main places it happens, and they have something called delayed developmental plasticity. And so this is where the dynamics get interesting. So what happens is that the environment experienced by the larvae affects what traits the adults have. So that's why it's called delayed, and it's developmental. And so here's the reaction norms that show, these have been fitted to data that show that relationship. So the amount of food per larvae per day that available is will affect the fecundity of the adults, and the average amount of food available per larvae also affects the chance that you survive the pupal stage to make it to an adult. So those are the two relationships that we're going to put into our model. So here's our model framework in terms of flow diagram. We have a larval stage. And the adults. I haven't explicitly modelled the eggs and pupae because I don't need to, because I just have to account for whether they survive through that stage, because they're not doing anything active. So that sort of gets absorbed, and we don't have to model that compartment explicitly. But with the larvae, we have a larval stage, and then these react according to what food the larvae are able to get, which is going to depend on population density, right? Because the more larvae that are competing for food, the less they're going to get. Those reaction norms then tell me what's the fecundity of the adults, and what's the likelihood that I actually survive to make it through to adult. So then we, according to this reaction norm, then we will then put the adults into the appropriate class, then according to their phenotype. So on the far left, those are adults that have experienced low food conditions as larvae. And so they have low fecundity, and they have a low chance of surviving to be from larvae to adults. And at the other extreme, these adults, that as larvae, they've had lots of food available to them, and as a consequence, they have high fecundity and a high chance that they made it from larvae to adults. So that's where the plasticity is coming in. And then we can pop that into the equations. I won't go into lots of detail, but it's the same structure I've already showed you. The equations for each life stage So there's the larvae, and then that's the Jth adult class. It's a mixture of recruitment, maturation, and death. Okay. And we can unpack that, we can parameterize all that. I just want to pick out a couple of things just to show you where that sort of trait and phenotype concepts are coming into the model. So, they're coming in various ways. So here's the adult recruitment. So that's the rate you come into here. So it's coming into the amount of food per larvae. The total amount of food's fixed, but the number of larvae is changing. So that's how the food per larvae is changing over time. It's affected by larval density. And that will determine this omega here, which basically just tells you it's a 0-1 function, this. So it just tells you, do I go into this class, or this class, based on the food? Okay. And then the other way it affects the recruitment into adults is through the reaction norm, the survival reaction norm that says, what's the chance I actually made it to adults? That's also affected by that food. And then if we look at the larval recruitment, the place that sort of phenotype is coming in again is here through the fecundity. So this term here is like the birth term of the larvae. And so if the larvae in the previous generation had lots of food, the adults are going to be big, they'll have high fecundity, we'll have a big queue, and then that feeds through the model. So that's how it's all coming into the model. Okay, and all of these things have been parameterized to lab data. Okay, so then we write, let's run the model. So we did that. And so I've zoomed, this axis is slightly different to the one you just saw, so I've zoomed in a bit. So, but the dashed line again is the data. So that's Nicholson's data that you've already seen. And the solid black line here is our model. And so you can see we're actually capturing the cycles really nicely. We're capturing that increase in population dynamics that we were seeing in the data. And we're seeing the oscillations stabilize as well. So it seems to be doing quite a good job, and it fits in with Nicholson's conjecture that it might be due to plasticity. I also want to just show you these coloured lines because we know exactly what proportion of the population have each phenotype. We can plot that out. So I've just done that for fecundity here, and that's what these coloured lines are. And the thing I want to show you here is that before, when food was high, we're seeing mainly yellow traits. And so that's quite a low fecundity. And then when food gets reduced, the adult food gets reduced, we're moving more towards the red-orange. And so we're seeing a shift in the trait distribution, and that's driving the dynamics that we're seeing. So we're now seeing individuals with higher fecundity. So that's kind of what's driven that pattern we're seeing, but we can unpack this a bit more. So the nice thing about doing these kind of dynamical models is that we can then kind of do experiments that haven't been done in the lab, and we can vary this adult food a lot more. So we did that. And so that's what I want to show you next. So this is what we found when we did that. So those people that have like kind of got a dynamical systems background, then you'll probably recognise this kind of plot as an example of hysteresis. But let me explain what this is. Those people that know a bit of ecology, then this is an example of tipping points. But let me talk you through it. So what we've done here is we've varied larval food, and then I've plotted the average larval population density long term, so as equilibria, and the same for the adult, but I've done the total adult, so I've summed up all those trait classes. And so what we see is, if I just walk you through one of these, is that as we change adult food, if we start with a small larval population, as we change adult food, the amount of larvae goes up until we get to this point here where basically this equilibrium doesn't exist anymore, and you get this sharp transition up to this other branch here, and we'll find our system now is mainly we now have a large larval population. If we look at the adults, it does the reverse. Okay, and so what's happening here? So this kind of jump up. This is because of bifurcations happened, but this jump here in ecological terms, people think about this as an example of a tipping point, where you've made a small change to the environment, here the adult food, but it's led to a large change in the population. And so then if you're thinking about ecologists looking at their data, they just change the food a bit and then suddenly their population dynamics have changed dramatically, and it's like, what's going on? So these kind of models can help us like unpick this kind of dynamic. So one thing I want to show you is that we're seeing this sort of pattern that I've already showed you in the data that was kind of not necessarily intuitive, is that low adult food, we were getting lots of adults, and high adult food, we're getting few, and they seem to have, larvae and adults have opposite patterns. So what's going on? We can actually use the model to understand this. And so what is happening is that on the upper branch for the larvae, so this branch here is the equivalent of this one in the adults, things are maturation limited. So when adult food is high, if we look at the flow diagram, what's happening? Adults' food is high, so then we produce lots of larvae. So lots of larvae, but then that's a lot of competition that's going on. So not much food per larvae. Not much food per larvae, there's a really slow chance that you make it to this stage. And so what happens then? So then you don't end up with many adults. And so the process that's limited is this maturation stage from here to here. So that's why we refer to it as maturation limited. It's the bottleneck. And so this high adult food is actually, you're not seeing the benefit of it because this competition stops you then realising that in your next generation. And then this bottom curve, which is equivalent to the top one here, the reverse is happening. So at low adult food, then low adult food, that means we don't produce many larvae, not much competition. High chance they'll make it through, and so then you get the higher adults. So that's why we're kind of seeing those patterns, and so it's really driven by the phenotype and that feedback. Remember that I was talking about, that we were introducing between the environment, the population, and the traits, really comes into play in these dynamics. So we were also able to do some analysis as well, and so. Even though it looks like quite a complicated model, we can actually, we were able to show that to get that S shape that I was just showing you, that hysteresis, those tipping points, that you needed to have the survival reaction norm, which is this one on the left, that needed to be convex somewhere in order for us to see this. So already we can say something ecological then about the types of reaction norms and how trait and environment relate by looking at the maths. And what I've plotted here is, this is the fecundity reaction norm, this is the survival one, and I've plotted a whole family of them, right? Because we can play with this. We're not constrained to the lab. We can play with different reaction norms and see what they do. And all the ones that are in red here are where we were actually seeing hysteresis in the model, so tipping points, and all the ones in blue were where we weren't. And so what's interesting here is that these points that I've got here correspond to where these points are. And so it's giving me an idea of the range of where in the reaction norm and in the environment am I seeing these tipping points happen. And the important thing is that they're not happening at the environmental extremes. They're not occurring right down here or right up the top. So what this means is that if we think about things from the point of view of environmental change, then when I'm changing my environment, classically what people think is that, okay, well, phenotypic plasticity should allow an organism to adapt to the environment. If the environment's changing, I can respond to that. That should be positive, and that is happening to a point. But then not anymore. And you might think, well, that's because now the organism can't adapt anymore. It's reached its limit in terms of how much it can change. But what this is telling us is that's not the case. So it's just because of this feedback that we're seeing that we're getting this more complicated dynamic. So that's just one example of where kind of thinking about these ideas about environmental change and how and plasticity and how organisms respond to it, we kind of get some interesting insights. But now I want to move on to a different example, and that's the mosquitoes. So this was really what motivated us to do the original work. We wanted a way of dealing with this problem. So the work I'm going to talk about is related to dengue transmission by mosquitoes. So mosquitoes are an example of a vector of disease, and what we've seen in recent years is the number of vector-borne diseases has gone up considerably over the last few decades, and within that, the number of cases of dengue and the number of countries affected by dengue, which is an example of a vector-borne disease, has gone up considerably too. And so mosquitoes vector some of the two biggest vector-borne diseases there are, which is malaria and dengue. So I got the reason we were interested in dengue was because we were interested in a mosquito that's present in Europe and is invading Europe. So it's called Aedes albopictus, and so you might have heard of it called the tiger mosquito. So that's what that is. It's got stripy legs. And what's interesting about that particular species was that basically people didn't think much about it in terms of transmitting dengue until 2014. And 2014, Guangzhou had a big outbreak of dengue. It lasted, I think, about 9 months. There was 14,000 people hospitalised due to it, and that was transmitted by Aedes albopictus, this mosquito. And this mosquito has been spreading through Europe and has increased its range. And what's interesting about it is it has evolved to now diapause. So what that means is overwinter. It's got a way of surviving our colder winters. And so that's enabled it to kind of get a foothold in Europe where some of the other species haven't been able to do that. And so there's been this real concern then about whether the diseases are going to come with it. And indeed, we have had cases of dengue locally transmitted in Europe by this mosquito. So in Italy, for example, and as recent as the Paris Olympics, there was cases of dengue locally transmitted as a consequence of this mosquito. So we wanted to understand, well, why are some places having these outbreaks and some not? The mosquito's it's got quite a big range in Europe, but there's these hotspots. What's going on? Can we explain it? So we wanted. So we're going to use our modelling framework there. There is. Ecologists are well aware that there's phenotypic plasticity and trait variation in mosquitoes and that it matters. So in mosquitoes, the thing that they often measure is the wing length of the adults. And that's a proxy for basically the size of the adults, and that corresponds to the fecundity and survival. So a bit like the examples I've seen before. So we can build that into our models here. It's another example of delayed developmental plasticity. So just like the blowfly, what happens is the environment, the larvae, and they're competing for resources, just like the blowfly were, and that affects basically how big the adults are going to be, the wing length of the adults, so therefore the fecundity and survival. So we could build all that in. You'll see that, so the adults, they're flying around, so they're in the air. The larval and egg stages and pupae, they're all in water. They live in aquatic environments. You'll see that I've got some extra boxes compared to the blowfly model, and that's because the mosquito has got various strategies for kind of protecting itself against environmental extremes. And so one example is that so this D box is it can diapause, so it can go into kind of stasis if it's cold. That's responding to by photoperiod and temperature. It also responds to precipitation, and so if it's very dry, it will stop developing and go into this quiescent state. So until conditions are good enough that it can, and it's wet enough that it can then continue development. So it's got all these ways of kind of buffering itself against changes in the environment. So we could put all this into a model, and that's what we've done. So what we've done with this model is we've parameterized all the ingredients using lab data. There's an awful lot of lab data out there. You have to do a lot of digging, and poor Ben and Dom spent many weeks doing this. But you can here's some examples of the reaction norms. So this one here is temperature and development rate of, I can't see without my glasses which stage, I think it's pupae. And so basically that's telling you like how long you spend in a life stage before you move on, and that varies with temperature. We could parameterize that, we fitted that. This is the survival of that stage. We can do that for all the life stages. There is data on that. These are two-dimensional reaction norms. So here we've got temperature and food affecting larval survival, and similarly here for the development of the larvae. But again, although I'm showing you the fitted function, we actually had fitted it to data. So we've used statistical models to fit these parts. So we fitted the individual gradients of the model, okay? So basically the functional forms in the equations, that's what we fitted. And then what we input into the model is the environmental data. So we use the climate data that we can download for whatever location we're interested in. So we can download temperature, photoperiods, precipitation data, and from that we can infer food data. And that is an ingredient into the model. So we've got our equations, we've got these functional forms that have been fitted to data, and then this input environment is then going to drive the model forward. So that's what we have. And then the output of the model is going to be, here's over time here, the population density and also the trait distribution. So here I've just done a plot of wing length, and the colours here are indicating all how much of the population of each wing length. So that's the kind of output. So I'm not fitting. I'm fitting functional forms, but I'm not fitting the output of the model to anything. So I want to make that point because I want to show you what happened when we then ran the model in a load of locations. So we wanted to validate the model. Is this any good? Is it even close to capturing the real data? And so here's the output of that. So so we run it globally. And so what we did, we took the temperature data for each location that we were looking at, and basically we scoured the literature for as much abundance data as we could get our hands on. And so you'll see the yellow here, yellow around the box is egg data, pink is adult data. We've got a bit of larvae data in the green, a little bit of purple is trait data, that's quite hard to get. But these are time series, so these are times on the x-axis, and so what we've got here is the blue is the model, and the sort of reddy orange curve is the data. But we've not fitted this. All we've done is just said, right, here's our model. Give me the temperature data and the environmental data for that location. Run the model, what comes out, and compare. And what you can see is actually we do a really good job of capturing what's going on. So we capture the seasonality. So and we can capture multiple years, and we can also capture multiple life stages as well, and across both temperate, so Europe, and more tropical environments as well. So we were pretty happy at this stage that we've got a model that was working. So now we were interested in ultimately in dengue, so we wanted to build that on. But before we moved on, we wanted to understand, well, what role has traits got to do with this? Can we say any? Is there anything we can say at this point before we move on? And we could say something. So one of the things that we picked out was kind of illustrated here. So typically, when people are doing kind of population models, they don't think about this kind of trait variation. And these feedbacks. And so what we did was let's run a model where all the adults in the population have the same wing length, because that's what people normally assume. Let's take it as the mean, but let's also just like try some different numbers. So that's what all these orangey yellow curves are here. They're versions of the model, but where adult wing length is fixed. So all the adults have to have the same wing length, irrespective of what happened to them as larvae, because that's what a lot of modelling assumes. And there are situations where it's not unreasonable when it does a decent job, but there are places like Guangzhou where it doesn't work very well. And here's why. So Guangzhou is tropical. And because it's warmer, the mosquitoes go through lots of generations, and so there's a lot of variation over time in the wing length. So this is the mean wing length, and this is the distribution in the blue there. And so because there's all this variation, when you compare it to models with fixed wing length, you start to see at the population level, the predictions aren't quite matching. So for example, The black line is our model, and you can see that we're saying, well, some years the mosquito will go almost to zero at winter, it'll go really low levels, and then some years you'll kind of see it pottering around still in winter. In these fixed wing length models, it's a sort of all or nothing. Either every year they'll be around in winter, or they won't, or every year they won't be. And there's sort of not this kind of mix. They also either tend to over or under predict the abundance as well. And so this trait information is somehow allowing us to capture some of the nuance of the dynamics that can't just be explained by just the temperature patterns. So that was kind of one sort of take home we had from looking at just the mosquito model. But then we thought, well, and this is, sorry, I should say this is why this is kind of thought flow of why. So fixed wing length model, let's just take an example. Suppose we have big wing length mosquitoes, right? Big wing length, big mosquito, lots of fecundity, lots of eggs. Lots of eggs, lots of larval competition, because lots of larvae fighting out for food. That means we're going to have a few adults, because not many will make it. And now the plastic model would then say that those should have a small wing length and low survival, but the non-plastic model would continue to have them as big and high fecundity. And that's where the things start breaking down if you don't include those feedbacks. So and that's what we're seeing here. So what about if we include, let's now extend it to include disease. So we're just sort of extending our framework. We have a human population that's split into susceptibles, infected and recovered. We introduce a further population of adult mosquitoes here that are infected. So if an adult mosquito that doesn't have dengue bites an infected human, then it can get the disease. And if that happens, then it's infectious and you can start this transmission cycle. So we could build all that into the model. We're also able to parameterize all that. So various of these processes are environmentally dependent, but we can parameterize all those functional forms that go in here. So then, and just as with the mosquito model, next step is let's compare it to as much data as we can. There's not huge amounts of time series on dengue outbreaks for Aedes albopictus, but this is what we found. And again, the blue is our model, and the orangey red colour is the data. And again, so we're not fitting that, but again, we see quite good comparisons. Where we're not doing well, so you'll see these sort of tails here. This is because in these locations there was actually control applied later on. And so we haven't got control in our model, any vector control in our model. So we're not going to capture these tails, but otherwise we're doing pretty well again of capturing the dynamics. So then with this model, we could then start looking at maps. So we then looked at, let's look at across all of Europe and see if we can identify where do we expect to get outbreaks. And so this is what we ended up with. So this map on the right is a map of average adult mosquito density. So you can kind of see it's everywhere. So a lot of predictions from a lot of models quite often end up predictions like this, that they find it's quite difficult to kind of isolate particular locations. And what we found is that with our model, we were actually the trait, including the trait dynamics, actually allowed us to somehow pinpoint more exactly where there were going to be hotspots, and that's what this map on the right is. So we could calculate something called a reproduction number. It's a bit like R0 that you would have heard of with COVID. So it's basically if it's bigger than 1, it says on average, like 1 infected mosquito leads to more than 1 new infected case. And so we looked at, so this map is saying how many months on average over this period, over an average year in this period is R0 above 1. And so we were able to pinpoint these hotspots. You see, most of the place, the disease is not going to transmit locally. So the infected mosquito has to come in. The infection has to come in from outside. Usually it's a human arriving with it. And then the mosquito has to bite them and then transmit locally. That's what we're investigating. Does that local transmission happen? And these little purple dots you're seeing is where our model's predicting, yes, it will. It's quite a small number of months it'll happen. It's quite a really narrow range of time, but it can. And the ones circled with the red here are where there actually have been outbreaks of locally transmitted dengue. And so we were able to predict pretty much most of the ones that, well, all of the ones that are in that have been documented, and some others. So and that might be that they haven't been tested, or just they might not been happened. We don't know. Importantly, we also picked we were also able to predict the one that happened in Paris this year. So we were quite when we tested that, we were quite it's quite sort of good that we were able to capture that as well. So somehow including this extra dynamics does somehow allow us to be a little bit more discerning in identifying where are those hotspots. And we could also see, well, what was making a hotspot a hotspot. And so I'm going to so the nice thing about these models is you can unpack it and try and understand it. So this is, I'm just going to talk about Guangzhou just because it sort of illustrates it most clearly. So this is time here. The blue here is the dengue cases predicted by the model, and this pink is the adult mosquito abundance. Now, what we did was we then looked at the model, and then we worked out, we looked at the infected adults and the ones that, and then all adults, and looked at their average wing length. And that's what these two curves are. So these are the infected ones in sort of the orange, and the green are just the average of all of the adult mosquitoes. And what this shows is that early on in the year, in the season, the most of the infection is in large adults. And what we found is that this seemed to be driving these outbreaks. And what's going on is that early on in the year, these large adults have a high chance of survival. So they live quite a long time. And so what happens is these then drive this initial takeoff in the infection. And then later on in the year, now it's warmed up. And so we tend to see shorter lived adults. And then so it's the smaller ones that take over later, and then we can see that here in the wing length size, take over later on. But what's driving that initial increase and that initial driver of the outbreak is actually only about 20% of the population, and it's those large infected individuals. And we were seeing that. What this graph here is showing you is, if we look at that's when the peak of transmission is. So we see here that the average wing length is much larger just prior to of infected individuals is much larger just prior to the outbreak, and we're seeing that in lots of different locations. So that was a pattern that seemed to come up again and again. So we could then start to sort of try and understand, okay, which mosquitoes are driving the dynamics. Okay, so I will just wrap up there. So hopefully I've kind of illustrated that how thinking about these kind of complex feedbacks in these systems can kind of start to reveal whether plasticity is actually going to be able to help these insects buffer the effects of environmental change. I mean, we also get these novel dynamics that lead to like interesting mathematical problems to solve, and we've been able to use this kind of approach to then try and understand some of the outbreak dynamics in dengue, and we're looking at trying to apply it to some other diseases as well. So I will stop there. Okay, thank you.
Well, thank you very much, Christina, for a great talk, and I think that really illustrates very well how mathematics can be used to understand complicated systems. So I have questions from the audience.
I realise that vertebrates and mammals and so on, particularly mammals and so on, don't have the same distinct sort of phenotype stages in that, but how much of that sort of model do you think would generalise to vertebrates and particularly mammals and possibly even people?
Yeah, Well, certainly, I mean, so for example, certainly like plants, they have life stages as well. So you could certainly use that approach there. With I guess with things like mammals, probably you wouldn't necessarily want to have the life stages, but the idea of the plasticity that happens. So I don't know whether you know if you know about snowshoe hares in Canada.
The dynamics with the.
Yeah, so snowshoe hares, they exhibit phenotypic plasticity. So in winter they're white, and they change their fur colour in spring to a kind of browny colour. They look more like hares we would see in the UK. And actually one thing that's happening, and you kind of see this, I was kind of seeing it in real life, like that they're getting their timing a bit off with the temperature. And so now you'll see kind of brown rabbits when it's snowy. And so they're, from a predation point of view, now they're in trouble because they're not doing so well at kind of responding to the environment and kind of using that plasticity to respond. So you definitely get these kind of ideas in other systems. You might, I'm not quite sure whether you want to use exactly the same framework, but maybe this idea of these feedbacks, I think that's probably relevant for other systems as well.
Questions, sir?
Really interesting talk. Thank you so much. I don't know much about the core model that you're talking about, so again, just like your question, maybe a bit naive. But you're modelling the population as a whole. So it's that everybody in your population at that time will have a certain trait. So how do you deal with the fact, because you had some delays in there?
Yeah.
How do you deal with the fact that actually the delays and the way, you know, that they aren't all laying eggs at the same time?
Yeah.
They aren't all doing, how do you deal with that kind of proportionality, or is it just a simplifying assumption?
No, it's a really good question. Let me just go back, let me get a flow diagram. So bigger one. Okay, yeah, so what's actually happening is though, so if I look at, if I just track one particular cohort, these individuals at this very particular moment in time will all have had, you know, got the same amount of food, and so we'll end up in a particular class. But very quickly the population, you end up with different cohorts coexisting because they've all developed at different rates. So what you're describing actually naturally comes out in the model. So at any one time, I've got individuals in all of these classes, and so what So what we're seeing here is actually the proportions in the different classes is varying over time. So if I plot these as distributions, you actually see changes in the distributions over time. Yeah, so that's in the model. What isn't in the model, there's other places of variation which I want to think about, which I haven't. So I haven't put the data on here, but basically I've just fitted the mean, right? But actually there's quite a lot of variation, and we know with a lot of these functions you tend to have more variation at the extremes. So one of the things I'm interested in is what if you think about that variation as well, and then what does that mean for all these kind of questions? So yeah.
Any other question?
Yeah, I've got another question from Nick, who we heard from earlier. I'll ask him if he's able to talk now. Are you there, Nick?
Yep, I'm here. Hopefully you can hear me.
Yeah, all good. Maybe introduce yourself as well.
Yeah, so my name is Nick Church. I'm the Principal Statistician for Environmental Science and Ecology with Biomathematics and Statistics Scotland. Really interesting talk. We have people in our organisation who do some pretty similar things in the kind of mathematical modelling area. So I've heard a little bit about this before. My actual question was about the distinction between individual phenotypic plasticity, population phenotypic plasticity, and gene cross environment interactions. So an example of an individual plasticity would be something like the example Rebecca talked about earlier, where trees that undergo a period of stress, once they've recovered from that period of stress, actually change their phenotype in response to that in order to be better to be able to adapt again. That's what I would consider phenotypic plasticity. It's an individual adjusting its phenotype in response to some stimulus. And that I don't think is in your model, in that so what I think your model is showing is population phenotypic plasticity. And if you would agree with that, how does that differ from gene cross environment interactions? How can you tell, are those two the same thing? Are they different things? Because the literature, there's quite a bit of fighting around whether gene cross environment or phenotypic plasticity are the same.
Yeah, okay, good question. I'm not sure I get this. You may well, I'm sure you know more about this than I do, so I'll give you my answer. So I think I'm going to try and argue that I am including individual plasticity, but this is where I'd be if you think that's not the case, I would be really interested to know. So the reason I think we're doing that is because of these reaction norms. So if you can see the slides, then these reaction norms are some sense are showing an individual response to the environment, and so we've got some environmental cue. And this graph would then tell us what trait we would expect that individual to have. What we're doing in the models then is tracking cohorts of individuals that have the same trait, so responding in the same way. I guess what I would take as the population plasticity, and so this is where I would really like to know your opinion on the terminology, whether I'm using it right. I would take that to mean when I start looking at the outputs, and if I looked at the distribution of traits in my population, that would be the population level plasticity and how that's changing to the environment. So if I then looked at the traits distribution at high food and at low food and compared, that would be me looking at how the population level phenotypic plasticity. So that's how I've been thinking about it. But yeah, if you think I'm not using that terminology right, then I would be really grateful to know that. So, yeah.
So I'm not sure I would agree that I would consider that individual phenotypic plasticity, because in order for that term to really be used, you would have to evolve you'd have to have a distribution in your reaction norms so that different individuals have slightly different relationships between their traits and their output. And then you would have to allow that to change over time in response to the life history of the individual. And that's, I think, the problem is that, well, not a problem, because I think the model clearly does brilliantly and it does a great job. But that's a sort of aspect that the model isn't capturing, is how the individual responds over time.
I think maybe I understand what you're saying. So yeah, maybe this actually comes back to what I was talking about when in answer to Rebecca's question. So yeah, so I'm assuming here that all individuals just do have the mean response, but what you're saying actually is that for a given genotype of that organism, that actually then if I look at any group of individuals with the same genotype, they're not all going to have exactly the same response, and that's that individual level.
Exactly.
Yes.
And
you're
then
absolutely right. We haven't got that. Yeah.
And then that in response, I mean, so we tried to put a grant in to study some of this, but we decided that you really need to track the life history of the individual too, and then evolve the reaction norm. If you were going to model it, for example, evolve the reaction norm in response to the life history, because for example, in Rebecca's example, the trees grow more roots because they have been stressed in the past, and so they change their phenotype so that they're not going to respond the same way again if exposed to the same stress.
Yeah, so we're ignoring any kind of genetic effects at all completely. So any change where the organisms, yeah, but we're basically assuming
it's a really interesting field.
Yeah. I would be interested to talk to you more about this. No, thank you. It's a really good question. Yeah.
Well, thank you. Any more questions? Oh, no more?
Actually, I have a question about the figure that you had showing the chart with the SIR model and the population dynamics.
So which
it had the SIR model.
Oh yeah.
I was just curious if because you mentioned wanting to sort of expand to looking at other statistics as well. When you do that, are you also thinking of using a different disease model depending on the disease that you're modeling, or is it preferable to kind of preserve the simplicities of the SIR model?
Very well. So we've done these kind of models for a few different mosquito and diseases, and so I very much would change it according to the system we want to look at. So, for example, we've looked at West Nile virus in Culex pipiens, which is a different mosquito that's present in the UK, and so that has what happens. The egg stage is a bit different. The humans are sort of a dead end host. It's birds, so we have things and sort of things are a little bit different how you parameterize. So you can use the same general approach, but you need to like building the biology for that particular system for sure. Yeah.
Okay, well, I think that's in the interest of time, we should stop here and thank Christina again.